I'm Dr. Denise Hurd, the Director of Research for the U.S. Poultry and Egg Association. Today, I have the pleasure of speaking with Dr. Nikki Shariat about one of her current research projects. Dr. Shariat is an assistant professor within the Department of Population Health at the Poultry Diagnostic and Research Center at the University of Georgia. Welcome, Dr. Shariat, and thank you so much for sharing your research with us today.
Hi, Dr. Hurd. Thank you so very much for the opportunity to speak today. We're really excited about this new project and so grateful for the U.S. Poultry Research Foundation for funding this work.
Thank you. Dr. Shariat, you recently started a research project funded by U.S. Poultry titled High Resolution Salmonella Stereotyping to Improve Surveillance in Turkeys. Can you tell us a little bit about this project and why there is a current need within the poultry industry for this type of research?
Sure. So yes, this is a brand new research project. We've just gotten this off the ground, and it's actually interesting. So usually as researchers, we usually give talks or interviews towards the end of a project, and we know what the results are going to be. And so this is new for me to talk before a project really gets started in earnest. So there have been a few high-profile Salmonella outbreaks in turkeys in recent years, and each of these have been attributed to different Salmonella serotypes. And as a result, there's been a big push in the turkey industry for improved Salmonella surveillance that's been supported both by U.S. Poultry and also by the National Turkey Federation. Unfortunately, traditional Salmonella surveillance is limited to only identifying the most abundant Salmonella serotype that's present in any given sample. And addressing this limitation is really key about what myself and my collaborators will be doing in this project. So we've always suspected that Salmonella exists in food animals as multi-serotypes, so not just a single serotype of Salmonella, but multiple serotypes. And work from my lab has shown this is sort of more often than not the case in broilers and also recently in cattle. And we expect that the same is also going to be true for turkeys. And the way that this problem manifests itself is that we can find certain serotypes in the processing plant, but we're unable to find those serotypes, and those are presumably ones that withstand processing interventions, but we're not able to find those same serotypes in live production. And what we're trying to determine in this project is whether those plant serotypes are actually present in live production, but that they're just masked or hidden by serotypes that are more abundant or better able to grow during live production. And to do this, as part of this project, we'll be collecting samples from turkey breeder flocks, from hatcheries, also from birds as they come into the processing plant, and then from different types of finished product. And then we'll profile for Salmonella serotype populations at each of these different stages. And the goal is that we'll be able to find that missing link between what we find in the plant and what we're able to find in production.
That's great, Nikki. And I understand that you are a huge proponent of sequencing technologies like the CRISPR-Seroseq. Will you share why and how CRISPR-Seroseq compares to other currently used methodologies?
Sure. Yes, thanks for this question. So CRISPR-Seroseq is a technique that we developed in my lab a few years ago. And to delve a little deeper into how the technology works, is it uses the sequence information from a specific part or region of the Salmonella genome, and that's the CRISPR region. And we know from work done by myself and others that CRISPR sequence is different in different serotypes. And that is to say that we can use that CRISPR sequence information to be able to serotype Salmonella. And researchers have been using this as a Salmonella serotyping tool for a number of years. What we've done with CRISPR-Seroseq is that we've sort of made this into a PCR and next generation sequencing assay where you can identify all of the different serotypes that are present in a sample based on their CRISPR profile. So this is a very sensitive approach, Denise. We're routinely able to find serotypes that are very low in the population, down to about 0.5% of the population. And if you were to contrast this with traditional sort of colony picking approaches, where you pick a colony off of the plate and you serotype this, those approaches only allow you to find the most abundant serotype. So, for example, if you had 2 serotypes present on a plate at 50-50, you'd have to pick 5 or 6 colonies to make sure that you got both of those serotypes. And if that ratio is 90% to 10%, you've got to pick about 32 colonies to find that. And so you can see that rapidly this sort of gets out of hand, and pretty quickly. And so being able to detect a serotype that's only 0.5% of a population is actually really powerful. Another methodology that's used to analyze and trace foodborne pathogens like Salmonella is whole genome sequencing. And when it comes to tracking outbreaks, whole genome sequencing has really been a game changer for food safety. Nonetheless, as powerful as whole genome sequencing is, it still relies on isolation of that single colony. So while you get a large amount of information about one Salmonella, you sort of still have the problem with only being able to find the most abundant Salmonella present.
Now, I believe that this study will focus on one particular company. Can you share how other producers might be able to take the findings and incorporate them into their own practices?
Sure, Denise. So for me, actually, this is the first time working in turkeys. Up until now, most of my work has been in broilers, and it's been really interesting to learn about the differences between these 2 different industries. And in particular, since turkeys aren't as integrated as broilers, I think our population analyses that we develop as part of this project can be really impactful for turkey producers. There's one specific application of this work that I think reaches across the poultry industry to turkeys and to broilers. And that's in the selection of Salmonella serotypes to develop autogenous vaccines. And this was actually the motivation for my industry partners in developing this project. So for autogenous vaccines, you survey the Salmonella that are present in your complex. So those that are present at the plant, and a greater weight, if you like, greater consideration is given to those serotypes that would be more commonly associated with foodborne illness. And then the next step is to go and check that you also find those serotypes in live production. And if you can't find them in live production, then you wouldn't necessarily use it as a vaccine, right? So it just wouldn't make sense because you don't see it in live production. So why vaccinate against it, even though you see it at the plant? But what if that serotype of interest does actually occur in live production, but at a much lower level than those other serotypes? And like I said earlier, Salmonella surveillance is a numbers game, so you only find those most abundant serotypes. And in this instance, you wouldn't find that one serotype in live production. And so with CRISPR-Seroseq, we'd be able to demonstrate that serotype did exist, and this might help in sort of better selection of Salmonella serotypes for autogenous vaccine development. And we hope that this would ultimately, contribute to mitigation of Salmonella across both industries. And so we're looking forward to starting to generate results from this project. And we're excited that its impact could extend to other turkey and broiler producers as well.
Absolutely. Well, I just want to thank you so much for sharing your work with us today. And before we go, is there anything else the industry should be excited to look forward to?
Sure. Thank you again, Dr. Hurd, for the invitation. This has been fun to talk to you today. When I'm presenting this work at conferences or meetings to my industry partners, I often apologize at this stage, right? Because on the face of it, it might seem that I've developed this tool that allows us to look for more Salmonella. And if you go looking for more Salmonella, you're going to find more types of Salmonella. And this mightn't be something that, on the face of it, people would be happy about. But in fact, I think that there's actually some key benefits to these types of analyses. And so one of those benefits was obviously in the development or the choice of serotype use for making autogenous vaccines. But this tool could also help to map Salmonella serotypes within a complex. And that could allow for example, for targeted biosafety measures to be implemented at certain points within a complex or certain farms. In addition, a more global benefit to these kind of approaches, and this is one that I'm very interested in myself, would be to involve national collaboration to routinely survey Salmonella serotype populations at pre-harvest, so that we can notice as soon as a different or a new type of serotype seems to be increasing within our industry. And And the ideal goal here is that this would give industry the benefit of time, right, to be able to be more proactive rather than reactive regarding these specific serotypes. You know, in an ideal world, this kind of approach might have helped us in, you know, in the broiler industry. We might have recognized serotype infantis before it became as big a problem as it had. And in turkeys, we might have recognized serotype redding before it became the big problem that it was. And I would love for our research, you know, through this project and other work that we're doing, to be able to contribute to preventing these industry-wide outbreaks in the future.
Excellent. And Nikki, thank you so much again. You have a lot going on over there on your side. And so I know myself as well as the industry are looking forward to the results of your research project. And we just thank you again for sharing with us just a little bit today about what you've been working on.
Thank you. Thanks, Denise. And we look forward to sharing those results with you. And thank you again to you and US Poultry, the Research Foundation, for supporting this work. We're truly grateful. Thank you.
Thank you.